Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we'll talk about gestation and olfaction. Gestation is the sense of taste. Taste begins with taste buds. These are located mostly on the tongue and within papillae. There are three types of papillae: fungiform, foliate, and circumvallate. Fungiform papillae are normally found in the front of the tongue. Foliate is found on the sides of the tongue, and circumvallate are found on the back of the tongue. Each taste bud contains multiple gustatory receptor cells, and each individual receptor cell responds to only one of the five basic tastes. The five basic tastes are sweet, umami, bitter, salty, and sour. For sweet, umami, and bitter, you have a GPCR receptor. For salty, you have a sodium ion channel, and for sour, you have a hydrogen ion channel. The labeled line theory suggests that each taste receptor cell has its own specific pathway to the brain, allowing the brain to determine taste quality based on which line is activated. Gustatory receptor cells synapse with afferent fibers of cranial nerves. So facial nerve 7 covers the anterior 2/3 of the tongue. The glossopharyngeal nerve 9 covers the posterior 1/3 of the tongue, and the vagus nerve, which is cranial nerve 10, covers the epiglottis and pharynx. So signals are relayed to the gustatory nucleus, then they go to the thalamus, then they go to the gustatory cortex in the insula. Now let's move on to olfaction. Olfaction is a sense of smell, and smell begins at the olfactory epithelium. This is where odorant molecules bind to specific olfactory receptors on sensory neurons. Each olfactory neuron expresses one type of receptor and sends signals to a corresponding glomerulus in the olfactory bulb. Olfactory receptors are GPCRs. Their binding triggers depolarization and an action potential. From the olfactory bulb, signals are relayed to the piriform cortex for odor identification, to the amygdala for your emotional response, to the entorhinal cortex for memory, and olfaction actually bypasses the thalamus, making it unique among sensory systems. Now we have a few theories of olfaction. So firstly, the label line theory also applies here, because specific receptors and axons encode specific odorants. Vibrational theory says that odorants vibrate at specific frequencies that receptors detect, but this is still controversial. And finally, we have steric theory. This is a lock and key model. So the shape of odorant determines whether it fits into a receptor and its structure-based activation. Next, we have anosmia. Anosmia is the partial or complete loss of smell, which can result from head trauma, viral infection, or neurodegeneration. Anosmia significantly affects flavor perception, which heavily relies on retronasal olfaction. You may have also heard that animals have pheromones. Pheromones are chemical signals that trigger innate behaviors in animals. This is detected by the vomeronasal organ, which is a VNO, and accessory olfactory bulb. Now, humans have a rudimentary VNO, but the system is likely non-functional. Effects of human pheromones are still debated, but we're not sure. To finish off, I just quickly want to go over the interaction between taste and smell. So flavor results from combined input from taste and smell. Volatile molecules from food travel through the retronasal pathway to the olfactory epithelium during chewing and swallowing. A loss of smell, like anosmia, drastically reduces perceived flavor. Overall, taste and smell are complex chemical senses involving multiple receptors, pathways, and brain regions. Understanding their detailed mechanisms and theories gives deeper insight into how molecules are transformed into conscious experiences. That's it for 6A. In the next episode, we'll go over some practice questions and then we'll start biochemistry.